Firstly, if there is any sort of emergency, we will hear first male voice. Thought about fruit flies before this evening. Didn't realise they were of major scientific interest and importance, but Tom's going to tell us all about it.
All right, thank you very much. So thanks everyone for coming. I hope this is interesting to you all. So as you can see, the title of my talk is Zipping Up Flies, and hopefully it'll become apparent what that title refers to, but I won't explain it quite yet. Right, so in this talk I'm going to be telling you something about embryonic development. I'm also going to be talking a little bit about wound healing, and I'm going to be telling you quite a lot about fruit flies, and in particular what fruit flies can tell us about embryonic development and wound healing. Okay, so let's start with embryonic development. So what is embryonic development? Well, embryonic development consists of the development of a recently fertilized egg, such as this, into a baby. And this is just a little movie here, which shows a sort of computerized representation of embryo development. Oops, embryo development happening. If my computer will wake up. No, it seems reluctant. Okay, we'll skip that. So embryonic development is really quite a remarkable process. So we're starting with something which is incredibly small and simple. So this is about the size of a speck of dust. 9 months, it's developing into a baby, which is an incredibly intricate and complicated being.
So how does this happen? This is a very interesting question. There's a lot of people around the world trying to understand this. It's also a very important question medically, because unfortunately embryonic development fairly often goes wrong. And if it goes wrong, this leads to birth defects. So around about 2 to 3% of babies are born with birth defects. And sadly, a large number of these will go on to die as a result of the defect. So I've listed some of the more common birth defects here. So Spina Bifida you may have heard of. And Spina Bifida occurs when the spinal cord doesn't form properly. And it results in a loss of control of the lower part of the body, and it can actually result in paralysis. There are a large number of congenital heart defects, and these can also be fatal. And you've probably also heard of cleft palate, and this shows a baby which is suffering from cleft palate and cleft lip. So these birth defects, they can be due to genetic causes, or they can be due to environmental factors. But for the most part, we don't really have a good understanding of what they're caused by. So this is one good reason why we like to try and study embryonic development. So there are ethical reasons why we don't study this in humans. So we tend to look at it in other organisms. And because it's such a complicated process, we try and look at it often in quite simple organisms where the process is somewhat more simple. And one of the organisms that we like to use is the fruit fly, which is also known as Drosophila melanogaster. And if you've not come across them before, then I've got some up here that you can come and have a look at later. So you may have seen fruit flies before buzzing around your fruit bowl in the summer. So their life cycle is rather similar to that of a butterfly. The life of a fruit fly starts when the female lays an egg. And inside this egg, the embryo develops over the course of 24 hours. Then the egg hatches and out emerges a larva, which you might know as a maggot. So this maggot then eats continuously for 4 days and massively increases its size. It then pupates, and then after another 4 days, a fly emerges from the pupa. So the stage that I'm interested in is this stage, the embryo. So, as I said, the female fly lays an egg. And when the female lays an egg, this essentially just contains a big bag of fluid. Over the course of 24 hours, this bag of fluid develops into an embryo. Develops into a larva, complete with everything that a larva needs to go about its business. So it has muscles it can move with, it has a digestive system, a nervous system, it can breathe. So that's a fairly remarkable transformation to happen over the course of 24 hours. All of these structures are formed just from a lot of fluid. So this is a very interesting question, which I'm trying to answer, is how does this all happen inside this little object? So I guess probably none of you will have ever come across a fly embryo before, so this is just to give you an idea of how big they are and what they look like. So this is a fly embryo, which I put on a 5p coin. The fly embryo is about 0.5 millimetre long, which is about half the height of the letters on a 5p. And that is pretty small. So that means that most of what I'm doing is down with some fairly powerful microscopes. Right, so why would we want to look at fly embryos if we're trying to understand human embryonic development? Because clearly a fly is very different from a human. Well, in fact, there are more parallels than you might think. So, for instance, flies have many of the same tissues that we'd find inside a human embryo, or we'd see developing the human embryo. So there are muscles, there are nerves, there's a respiratory system, a digestive system. And although these are different to the ones found in us, they're a kind of simplified form of the same thing. So it gives us a good starting point to try and understand these structures. So another interesting thing is that the development of all these tissues is actually controlled in a very similar way in flies to how it is in us. And a good example of this is the development of the heart. So the fly larva has a form of simple heart that pumps fluid around the inside of the larva. So some years ago, some scientists were studying a newly found gene in flies, and they found that if this gene was mutated, the larva no longer had a heart. So they called this gene Tin Man, after the character in Wizard of Oz that doesn't have a heart. And so it turns out this gene is very important in controlling the development of the heart. So a couple of years later, some people were working on human genetics, and they were surprised to find that actually humans also have a Tin Man gene. And amazingly enough, this Tin Man gene is also required for human heart development. And they found that certain sufferers from congenital heart defects were suffering because they had mutations in this Tin Man gene. So this is quite a good example of how we can use flies to learn more about human genetics and about human diseases. Right, so why else would we want to study flies? Well, so as I said, their embryonic development takes place over a very short time scale. So this means that they're quite amenable to work with in the lab. This is far easier to work with than something that takes 9 months to develop. So we've already got a good understanding of fly genetics because people have been looking at fly genetics for about 100 years now, so we know an awful lot about fly genes. And we're actually quite good at being able to manipulate these genetics. So the other thing that's good about flies is that we can easily watch their development happening. So the embryo is small and it's transparent, which means that we can actually watch in real time, we can watch the embryo developing. So just to give you an example of this, this is an embryo. Rub the lights down a little bit. Thanks. This is an embryo in which a particular type of cell called a hemocyte has been labelled with a fluorescent protein. And this allows us to watch how this cell moves through the embryo during development. So these cells are called hemocytes, and they're the equivalent of white blood cells. So their job is to fight infections inside the fly embryo. And if I play this movie, we can see how these cells move around inside the embryo during the development. So this is something, there's no way we could do this in a human embryo or a mouse embryo or something like that. We could never look at something with this much detail. Right, so I'm going to be talking quite a lot about using fluorescent proteins to follow developmental processes in this talk. So I thought I'd just give you an idea of how we go about making fluorescent embryos like this. So it all starts with a jellyfish. This is not a normal jellyfish, this is quite a special one because it's able to produce green fluorescent light. And it can do this because it's got a protein in it called green fluorescent protein, or GFP. So what we're able to do is take the gene which encodes this protein from the jellyfish and inject it into our fly embryos. So if, with a bit of luck, this gene will incorporate into the fly's genome alongside all the fly's normal genes. This embryo can then develop into an adult fly, and when this fly has offspring, it will give rise to green fluorescent embryos. So these look very nice, but actually they're not very useful because, as you can see, these are green all over. So what's more useful is if we can label specific parts of the embryo and really understand how they're developing. So like the movie I just showed you where we looked at the hemocytes, we can do that with this by modifying this gene that we add. So we can make it just, we can make this gene just active in particular cell types. So for instance, in the hemocytes, we can make it so that it was only active in nerves, so we're only labeling the nerves, or muscles. So we can basically choose which process we're interested in, and we can really follow it in real time and a lot of detail in the development. So we can also do the same trick with a red fluorescent protein and make red embryos. And if we cross together these two flies that have the red and the green genes, we can make one with both, and they're yellow embryos. And obviously these will hatch into red, green and yellow larvae. And here's some of these. And I'll just play this movie so you can get an idea of what they look like. So here's some fly larvae crawling around in the lab on a petri dish. So this may look nice, but there's a proper scientific reason for doing this, which I'll come back to a bit later. But that gives you an introduction to the kind of tools that I'm trying to use. Right, so to get back to development. So the particular developmental problem that I'm trying to investigate is how we join edges together during embryonic development. And the reason I'm interested in this is that this often fails and leads to birth defects. So what do I mean by joining edges together? So if you imagine that we're trying to make a tissue, which is kind of like a sheet, so something like skin, then often in an embryo we'll make it in small sections, then we have to join them together. So that's an example of this. So we're taking two sections of skin and we're fusing them together. So another time is if we want to make a tube. So often when we want to make a tube in an embryo, we start with something flat and we roll it over, join the edges together. And I'll give you an example of that in a minute. So there's a couple of things to note about doing this. So these processes have to occur completely. So what I mean by this is if you're making skin, then it needs to be a protection against the outside world, and you can't have holes in your skin, otherwise you'll let bacteria get in. So we really have to make sure that this is happening very, very well. And it also has to happen accurately. So what I mean by this is that we have to be sure that this piece of skin is meeting a piece of skin. It's not bumping into a bit of heart and fusing with that, or a bit of liver. We need to be really sure that it's skin fusing with skin. So there's actually quite a lot to this process. So I'm going to give you an example of a joining event or a fusion event like this in human development. And that is called neural tube closure. So the neural tube is a structure which will eventually go on to become the brain and the spinal cord. And the neural tube starts life as a cleft, not a tube. And what happens is the two edges of the cleft join together and fuse in the middle of the top of the cleft. So you can see this is a neural tube, a human neural tube, which is just in the process of fusing. So you can see this central portion is fused, but this bit at the bottom still hasn't come together, and the bit at the top hasn't come together. So what's important about this? Well, the importance of this is that it's this process that fails and leads to spina bifida. So if this is the tail end of the neural tubes, this will become the spinal cord. So if this fails to fuse, then it will lead to the baby suffering from spina bifida. If this end, which becomes the brain, fails to fuse, it actually results in a more serious condition called anencephaly, and this is always fatal, unfortunately. So this is quite an important problem to try and solve. Try and understand why this failure is happening. And I also should note that there are actually a number of other defects, birth defects, that result from a failure of these kind of fusion events. So another one is cleft palate. So cleft palate results from a failure to fuse two edges together. So as I said before, I'm using the fruit fly to try and understand how situations like this occur. So if we can get a better understanding of how these fusion events really happen, then it should put us in a better position to understand what's going wrong in spina bifida and put us in a better position to maybe try and prevent that from happening or to repair it once it has happened. So Drosophila embryos have a fusion event of their own. So just like us, the Drosophila larva needs a skin to protect it from the outside world. And this skin is called an epithelium in this case. So, so this skin or epithelium starts to form actually fairly early in embryonic development. But up until around halfway through embryonic development, there's a big hole in the middle of the skin. And the process I'm looking at, which is called dorsal closure, is when this hole is filled over. So this is an embryo, and this is the skin on one side, this is the skin on the other, and this is the hole that we're trying to cover over. And I'll just play this movie, and you can watch this, this happening. Okay, so we see the skin moving over the embryo and fusing when it comes together in the middle. And once it's finished, we could never tell there was a gap there. It fuses perfectly. So we can get a much better idea of what's actually happening as these two bits of skin fuse by looking at an embryo which has got green fluorescent protein in the epithelium. And what we see is that around the edge of the hole, so along the two edges, and we see this kind of concentration of green. So we call this an actin cable, and the job of this is to keep the two edges nice and organised while the fusion's happening. And more importantly, we can see coming into the hole these kind of needle-like projections. When I play this movie, you can see, hopefully you can see what the job of these little projections is. So I'm going to play this, you can see these things coming out, and they seem to grab the other side. And they kind of knit together the two bits of skin. So there we go. So that's telling us something about how these fusion events happen. So this kind of joining together with these kind of needle structures has been called zippering. And I think you can see why it's called that in this next movie I'm going to play. So this is a slightly different fluorescent protein. So this really nicely labels these sort of needle-like projections, so we can really see what they're doing. And I'll play this movie, and you can, and I've put some zips at the bottom just for comparison, and hopefully you can see why we call this zippering, this process. So if you look at these ends, it really does look like a zip closing. So it really looks like that's how embryos join edges together, they zip them together. But is this just something about flies, or does this happen in other situations as well? Well, we think that actually this is probably the way that all these junctions happen in embryos. So here's an example from a mouse. So you may not know this, but mice are actually born blind, and this is because during their embryonic development, their eyelids fuse shut, and they don't open again until about 10 days after they were born. And if we look with an electron microscope at the eyelids as they're fusing, we can see just the same thing that we saw in the fly embryo. So we see these same needle-like projections interconnecting with each other. So we think that this is fusing by exactly the same mechanism that we saw in the fly. So what's really nice about looking at this in fly is that we can really start to get to grips with what the machinery is that is controlling this fusion process. And this is where using two different colours of fluorescent protein comes in. So now I've labelled one protein in red, and this nicely labels these needle-like projections, and another one in green. So this protein in green is a protein called ENA. And as you can see, it's found just at the tips of these little projections. And the job of ENA is to make these projections. So without ENA, we can't make these. So we can now watch, if I play this movie, we can watch what we call the dynamics of this ENA protein during the process as this fusion happens. We can see it always on the end of these projections. So the interesting thing about ENA is that it's not just flies that have ENA. We have an ENA protein too, and so do mice. And interestingly, some other scientists in America have recently been looking at a mutant mice which can't make this ENA protein. So this is what an embryo from this ENA mouse looks like compared to a normal embryo. So what you can probably notice is that this mouse that doesn't have any ENA hasn't managed to close its eyes. So presumably this is because it can't make these projections, it can't do this kind of fusion process. So it also has this structure up here. So this is an exencephaly, and this results from a failure to close the neural tube. So this is similar to Spina Bifida. And in fact, this embryo has lots of problems associated with failure to join edges. So we think this is telling us that in embryos, we always join edges in just this way, but it's only by looking at flies that we could really watch this happening and really understand how it works. Right, so now something that's important to realise is that embryonic development has to happen in a very precise manner. And this is true for flies just as it is for us. So if you've ever looked at a maggot, you might have noticed that it's made up of segments. So this is an embryo which has just finished dorsal closure, and you can see these segments have formed already. And actually these segments form before dorsal closure, so we can see this embryo here also has these segments. So this presents the embryo with a bit of a problem, because it means that when it brings the two bits of skin together, it has to bring them together absolutely perfectly. Otherwise, the segments on one side won't match the segments on the other, and you'll end up with a kind of misshapen larva. So I was interested in trying to understand how it closes this hole just quite so accurately. So we've got this useful fly for looking at this. So now, again, we're expressing green fluorescent protein in the skin, but now the gene is only turned on in the back half of each segment. So that means we get these stripes. I'll play this movie, and what we find when we look at this stripy embryo is that the stripes from one side always come together perfectly with the stripes on the other side. So this is showing us that this is actually a very accurate process, and we want to try and understand how the fly can achieve this sort of level of accuracy when joining these two edges together. So to try and get to grips with what's happening here, I decided to make this new fly. So this nice stripy red and green fly, and the two sides should be symmetrical. And what this means is that if the two sides are coming together perfectly, we should always get the same colour matching the same colour. And this should allow us to see what would happen if two different colours meet. So what happens? Because that would cause a mismatch. And we could look at this and try and understand how the fly deals with this. How does it deal with the situation if it's not matching perfectly? So this is just a little short movie which just shows just part of the hole, the end of the hole. And what you can see here is that these red cells here, so this red stripe has to match with this red stripe, but it's not very well aligned at the moment. And what we'll see happening is that these projections search this opposing bit of epithelium, but they have shown no interest in fusing until they get close enough to these red ones. So I'll play this and you'll see this happening. So we see these red projections searching along this other epithelium, but they don't want to attach, and they pull back, and they search again, and then eventually they reach as far as the red stripe, and then they rapidly fuse with it. So this is telling us a couple of things. So firstly, it's telling us that these projections actually have two jobs. So we need them to stitch together these two sheets of cells, and they also play a job of kind of searching for the correctly matching cells. So they can search ahead of these cells and try and find the correctly matching cells. So the other thing it's telling us is that actually this is a very accurate process. So not only are these cells able to recognise the tissue they're supposed to attach to, which is this epithelial tissue, the skin, they're also able to recognise exactly which cells in that skin they need to fuse with. So, so I think that maybe this is perhaps giving us new information about what might be going wrong in something like Spina Bifida. So maybe in Spina Bifida there's a similar, or it's probable in Spina Bifida, there's similar recognition events that make sure that the neural tube is fusing in the right place. So maybe in Spina Bifida we've lost the ability to recognise the cells when we come across them. So what I'm trying to do now is to isolate genes which are involved in this recognition process. And if we can find what these are, then this might provide us with a starting point to try and understand the genetics of conditions such as Spina Bifida. Right, so far I've been talking about embryonic development. But there's actually a similar fusion event that probably happens to all of us from time to time. And that is when we need to heal a wound. So we started to wonder, well, this looks like wound healing, so maybe it's not just superficially similar. Maybe there really is something very similar to what's happening in embryonic development and what's happening when we heal a wound. So we started trying to make wounds in the fly embryo so that we could compare what happened when these wounds healed to what happens in this process. So this presents us with a problem. How do you wound something which is microscopically small? So it turned out that the best way to do it is with the laser. So we can get a very accurate laser that can make a very small hole in the fly's epithelium, such as this. And what we found when we made these holes was that it was very similar to dorsal closure. We saw this kind of cable around the edge of the hole, and we saw these sort of needle-like projections. And as I play this movie, what you should see is that it does look very much like dorsal closure. So we see these projections joining up across the hole and sealing the two sides of the hole together. And subsequently we found that a lot of the genes which we need for dorsal closure, we also need to close wounds. So this is what this is telling us, is that when we need to heal a wound, our bodies just turn on the same set of equipment that it previously used during embryonic development. And so by studying these kind of fusion processes in development, we've actually been learning new things about wounds. So we wanted to try and think, well, maybe what we've learned, maybe we could try and improve how we heal wounds. So this is a scene from the film X-Men, and this gives you an idea of what we might be trying to achieve with this sort of thing. So as you can see, this is Wolverine, if anyone's seen the film will know, and at the moment he's got rather a nasty wound on his face. And as I play this movie, watch what happens to the wound. So you can see it's already looking a bit smaller, shrinking back. And then it's moving on. And then if you look right at the end of the film, what we'll see is that it's totally gone, and it looks perfectly back to normal. So Wolverine's managed to do two very clever things here. So firstly, he's healed up his wound very quickly, and he's also managed to heal out without any scarring whatsoever. So we'd like to try and achieve this. I think maybe it's a bit unrealistic to think that we could ever do this, but maybe we could get somewhere. I think there's room for improvement. So what I'm going to talk about now is how we might reduce scarring. So you may not think that scarring is a big problem, but actually it can be. So often scar tissue can cause more damage than the original wound did. And this is particularly a problem after surgery. So often a surgical procedure will result in the formation of a scar, which can cause a lot of complications. So it would be very useful if we could somehow learn how to reduce scarring in some way. So how might we go about this? Well, we could look for inspiration in the sun, maybe. So on the front of the sun here, there's a story about a baby who was stabbed while he was in the womb, and miraculously this baby managed to survive. But there's actually something else which is quite miraculous about this baby, and also other babies that suffer from these similar kind of bizarre events, is that when they're born, they have very little sign that they're ever injured. They're somehow able to heal their wounds with very minimal scarring. And in fact, recently, surgeons have started operating on embryos while they're still in the womb. And the same happens here. If the surgery is successful, when the baby's born, there's not much sign that the baby was ever operated on. So there is something about embryos that allows them to be able to heal wounds without scars. So, and if we could somehow understand what this was and harness it, maybe we could use it to make us be able to heal wounds without scarring. So what is it different about embryos and us? So we can perhaps get a clue by looking at a particular strange mouse mutant, which is called the Pu1 null. So just like embryos, this mouse is able to heal wounds with very little scarring. So is this some kind of amazing super mouse? Well, unfortunately it's not, because these mice also die very young of septicemia. So why do they die of septicemia, and what's this got to do with their ability to heal wounds without scarring? Well, what's different about these mice is that they can't produce two kinds of white blood cells called neutrophils and macrophages. So the job of neutrophils and macrophages is to fight infections. So the job of them is to eat up any bacteria that get into our bodies. So they're an essential part of our immune system. And that's why these mice die so young. They can't protect themselves against infection. Okay, so what's this got to do with wounding? Well, to explain that, I have to explain something called the inflammatory response. So, when we sustain a wound, neutrophils and macrophages, so they're the two cells that Pu1 mice don't have, leave the bloodstream, they move through our tissues, and they move to the wound. And their job there is to eat up all the bacteria, any bacteria that have got in, and also to clear up the sort of general debris which has resulted from the wound occurring. So what's different about Pu1 null mice is they don't have these two types of cells, so they can't mount an inflammatory response, so they don't have these cells going to their wounds. So maybe this has got something to do with their ability to not produce scars. Maybe there's something about these cells going to the wound which leads to scarring. So a bit of evidence that supports this is that what these mice have in common with embryos is that embryos also don't produce an inflammatory response. Okay, so there are various other bits of evidence that suggest that maybe there is really a link between these two types of cells coming to a wound and scars forming. So what we would really like to do now is to try and see what would happen if we could prevent these cells going to wounds. So could we maybe find a way of preventing scarring by blocking the inflammatory response, blocking these cells going to the wound. So the trouble at the moment is that we really don't know what attracts these cells to the wounds. We don't know what's making them leave the bloodstream and go to the wound. So we'd like to try and understand what it is that attracts these cells to the wound. So once again, we'd like to try and do this using fruit flies. So as I said before, fruit flies have a cell type called hemocyte. So this is the fly's kind of equivalent of a macrophage or a neutrophil. So if we wound the fly embryo, so there's a big wound here, then these hemocytes will move to the wound, and I'll play this movie, you can see this happening. And their job is to, again, just like in us, is to clean up debris and to prevent any infections from getting in this wound before it's closed. So we can really see how these cells clump up around the edge of this wound. So this is quite a nice system because in us, in humans, we can't watch this happening, but in flies, we can really watch this inflammatory response happening. So at the moment, we're trying to identify genes which are involved in attracting these cells to the wound. So we're trying to look for fly mutants in which these cells don't go to the wound, basically. And maybe this will provide us a starting point for trying to block this from happening in people, and maybe this would be a way of reducing scarring. So just before I finish, I wanted to talk about another animal, leave flies behind for a while, because maybe flies aren't the best system for looking at this in. And we've got a new animal we're using in the lab, which is perhaps a bit better. This animal is the zebrafish. So this is a small tropical fish. And this is what the zebrafish embryo looks like. So it's actually just a couple of millimeters long, so it's pretty small. So the advantage of zebrafish over flies is that zebrafish are much more like us in some ways. So their macrophages and neutrophils are very much like our own macrophages and neutrophils. And also they have blood circulation. So this means that the inflammatory response in a zebrafish is really quite similar to our own. And the advantage of looking at zebrafish is that we can watch this inflammatory response happening. So just the last thing I'm going to show is this movie of macrophages and neutrophils leaving the circulation and moving to this, which is a wound which has been made in the fin. And you should see these cells kind of crawling their way through the fin to the wound when I play this. So this is very speeded up, so you can see that the blood's flowing very quickly in this vein. But you should see these cells sneaking out and going to the wound. Yeah, and this is something that we really can't do in humans, can't do in mice. So this is really a very nice system to be able to really follow this important process happening. So we're very hopeful that we can find out something really good information from this work. So maybe I could come back another time and tell you what we found. So that's all I'm going to say for today, but I should just thank a few people. So all this work's been done in the lab of Paul Martin by various people, not just me. So I should thank Brian Straver, Sarah Warner, Antonio Jacinto, and Will Wood.
It's work done at Bristol University and financed by the Wellcome Trust. So thank you very much. Thank you for listening to me. And I've got some flies if you want to look at flies here.